The next thing we're going to talk about here as it relates to the cell itself is what's usually referred to as the cell life cycle. The cell life cycle is based on two distinct things. First thing is what's referred to as the genetic machinery. The genetic machinery is the ability for the cell to make proteins to do things so that it can regulate the cell's homeostasis. The second part of the cell life cycle is making new cells based on the cell's genetic machinery not being able to meet the demands placed on the cell. Within this cell life cycle, we have this distinct sequence of events referred to as the genetic machinery. In general biology, we refer to the genetic machinery as the protein synthesis cycle. However, we have to add something into this, and that something into this is a response to a stimulus. The response to the stimulus triggers an intracellular activation sequence. This intracellular activation sequence leads to the transcription of DNA into its component RNA sequences, where we were going to take the double helix, splice it apart, replicate one of the strands in order to make an RNA sequence strand. That RNA sequence strand is then going to be able to exit the nucleus, the nuclear pore, interact with ribosomes to allow for translation of the RNA into its amino acid sequence or the polypeptide chain. That polypeptide chain, based off of enzymes being turned on or enzymes being turned off within the cell itself, will go about a process known as post-translational modification. That's where we take a peptide chain and make it into a functional protein. This functional protein leads to a functional change, and this functional change is hopefully enough change so as to inhibit the stimulus that the cell was initially being exposed to. The ability for the cell to meet the functional change is going to dictate how long is the cell going to go through its normal life cycle. That is, its normal metabolic processes to do its normal functioning. Once it's unable to make enough functional change to meet the demands of the stimulus, it will then trigger the next sequence of events within what is referred to as the cell's life cycle or the cell cycle itself. So let's talk about the cell and the cell cycle itself. That is, how is the cell going to go about replicating itself? When we talk about the cell cycle, we basically break it up into distinct phases. These distinct phases start with what's usually referred to as the G1 phase, that's the growth phase, followed by an S phase, which is the synthesis phase. In the synthesis phase, we're going to go about replicating the genetic information, the DNA, in which all chromosomes will go about a full replication so that the daughter cells coming away from the cytokinesis will allow for two new functional cells. The S phase is followed by the G2 phase, and the G2 phase leads into mitosis, which is prophase, metaphase, anaphase, telophase. And then finally, we get the cytokinetic process, which is embedded into mitosis, but is not actually part of mitosis itself. However, there is another phase that comes into play here within cells of the body, and that's what's referred to as the G0 phase. The G0 phase is the phase of the cell cycle where the cell is doing its normal functioning. It's not undergoing distinct growth factors, which will cause the cell to become larger. It's simply doing what the cell is supposed to be doing. And what ends up happening is that the cell within the body can be bouncing in between the G1 phase, that is the growth of the cell, and the G0 phase, that is the normal function of the cell. Most cells will be bouncing back and forth between this G1 and this G0 phase for most of the cell's life. It's not until the cell becomes too large for what it needs to do or where it needs to go about a replication so as to respond to some sort of injury that we will go ahead and move into the rest of the cell cycle. There are some cells within the body that will remain within G0 for most of your lifespan. These include things like skeletal muscle, cardiac muscle, nerves, neurons. things that we usually reference as being amitotic. Those are cells that do not do division. So when does the cell know it's time to divide, and when does the cell know when it's time to stop dividing? So let's take a look at these two questions in this animation.
Scientists developed two experiments. The first was to apply cultures of cells on nutrient medium in petri dish, and the cells continued to divide until they filled the dish. Then an injury was inflicted by scraping away some cells down the middle. You might consider then that the cells along the margin of the injury began to divide until the cell petri dish was filled again. Based on the experimental results, you might suggest that the controls for the cell cycle were external in origin, meaning that cells, when they come in contact with each other, scientists developed two experiments. The first was to apply cultures of cells on nutrient medium in petri dish, and the cells continued to divide until they filled the dish. Then an injury was inflicted by scraping away some cells down the middle. You might consider then that the cells along the margin of the injury began to divide until the cell petri dish was filled again. Based on the experimental results, you might suggest that the controls for the cell cycle were external in origin, meaning that cells, when they come in contact with each other, stop dividing. This is called contact inhibition. But a second experiment, one that involved removing cytoplasm from a cell in metaphase, really changes this perspective as well, because that cytoplasm was then injected into a different cell that was in interphase, and with that injection, the cell began to initiate a new mitotic process. So in this case, we see that the cell cycle is controlled by internal controls, or internal regulation, and this is by protein compounds referred to as cyclins, and they control checkpoints along the cell cycle. There are three checkpoints we're most interested in. The first is called the G1 checkpoint, and it's concerned with evaluating the two questions. Is the cell big enough, and is the environment suitable, or is there space available? Think of this as a lot like a traffic control signal. Cells that can answer these questions in the affirmative move on to the next stage. If they cannot, then they stop and go back to G1, where they may continue to grow or continue to perform their cell activities. Some cells remain in the G0 stage for their entire existence. The second checkpoint is called the G2 checkpoint, where DNA is concerned with. The next checkpoint is called the G2 checkpoint, which is primarily concerned with DNA being copied correctly. The third checkpoint is called the metaphase.
So if we look at the table that we see here, well, we notice that there are some cells that are excessively long-lived, and there are some cells that are excessively short-lived. If we look at this table, what we can think about is what types of cells will we most expect to see cancers coming from, and what types of cells will we least expect to see cancers coming from. So if we think about this, the neuron does not really divide, which means that what cells are causing things like brain cancers to come from? Whereas there are other cells, such as, say, the red blood cells or the erythrocytes, which are relatively moderate lived, but the problem with the erythrocytes is that they are metabolically dead once they are fully derived. Whereas other cells, such as cells that are lining the lumens of the esophagus or lining the lumens of the small intestines, are constantly undergoing divisions because of the exposure that they have to all of the things that they are interacting with and the high demand of metabolic activity to either absorb or secrete materials based off of those interactions. And so when we talk about the tissues here in a second, what you have to think about is what tissues are going to be within the G0 phase for extended periods of times relative to those that are going to be constantly undergoing cycling. And the constant rate of cycling is going to dictate what the overall ability for those cells to adapt will be. And so when we look at the adaptations that might take place within certain tissues, cells that are very long-lived will have large amounts of adaptations taking place, whereas cells that are short-lived tend to have very little adaptations that take place.